Hello everyone, I'm Anas Al-Ubaidi, and the title or the topic for this lecture or session is Investigational Product IP for short, Development Lifecycle, which is part of the chapter of Introduction to Clinical Research and Regulatory Frameworks. And of course, this lecture is part of the Advanced ICH Good Clinical Practice Certification Course Program by CCRPS. Introduction. The IP, the investigational product, sometimes also referred to as SM, which is short for study medication, is a drug, biologic, or device under investigation in clinical trials. So it could be a device or could be a medication, a drug. The IP development cycle or life cycle refers to the comprehensive process of creating, testing, and obtaining regulatory approval for an IP, investigational product. This life cycle is complex and involves multiple stages, each crucial for ensuring the product's safety, efficacy, and compliance with regulatory requirements. Key points. We'll start with initial discovery and preclinical research. Preclinical research, this phase involves laboratory and animal testing to assess the IPs, the investigational product safety and biological activity. Objective: evaluate potential toxicity, dosing, and side effects. Example: testing on animals to determine the pharmacokinetics, how the drug is absorbed, distributed, metabolized, and excreted. So, testing all four processes of PK: pharmacokinetic, which is absorption, ADME, which is short for or acronym for absorption, distribution, metabolism, and excretion. Formulation: development of the actual formulation of the IP, which may include drugs, biologics, or devices. This stage ensures the IP is stable, bioavailable, and capable of achieving therapeutic goals. Example: a pharmaceutical company develops a new oral cancer drug and performs preclinical tests to understand its effects on tumor cells in animal models. Another key point here, IND, investigational new drug application. Before clinical trials begin, an IND, investigational new drug application, must be submitted to the regulatory body, for example, FDA in USA, for approval. Of course, this applies in general to all regulatory bodies around the world. Let's say EMA in Europe, European Union, Health Canada in Canada and such, or PMDA in Japan, or MHRA in United Kingdom, in UK. But maybe in different names. The application will be in different titles, under different titles or names, but similar to IND in FDA. In FDA, it's IND. So what is an IND? Investigational new drug application. A request to the regulatory authority for permission to start human clinical trials. Requirements here: preclinical data, animal studies, manufacturing information, how the IP is produced, clinical trial protocols, plan for human studies. Requirements: so the preclinical data, the tests that occurred or happened or conducted on animals, the manufacturing information, how the IP is produced, and the study protocol or the trial protocol. Example: a company submits IND to the FDA to begin phase 1 clinical trials for a new drug that targets a specific type of cancer. Another key point here: clinical trials, phases 1 to 3. Clinical trials are designed to test the safety, efficacy, and dosing of the IP in humans. The process is split into several phases. Phase 1, objective here: test safety, determine dosage range, and identify side effects. Participants: small number of healthy volunteers. Example: testing a new oral drug on or medication on 20 healthy participants to determine the correct dosages and identify any immediate side effects. Phase 2 objective here will be evaluate the efficacy of the IP and further assess safety. Participants, also a small group of patients with a target with the only difference than phase 1 with the targeted conditions, not healthy participants, but like in phase 1, but in phase 2 targeted with the targeted condition. Example, administering the drug to 100 cancer patients to assess its ability to shrink tumors. Phase 3 objective here, confirm efficacy, monitor side effects, and compare with existing treatments. Participants here, a large group of patients. Example, conducting a large trial with 1,000 patients to confirm that the new cancer drug is more effective than the current standard of care. And here's a diagram on clinical trial phases sequence. Another key point here, new drug application, NDA, or biologics license application, BLA. So BLA, biologics license application, or NDA, new drug application. Once clinical trials demonstrate the IPs, or investigation product safety and efficacy, the sponsor can submit an NDA, new drug application for drugs, or BLA for biologics, to seek regulatory approval for marketing for phase 4 in order to conduct phase 4. So between phase 3 and 4, we have NDA. So before phase 1, we have IND, new investigational new drug application. NDA, new drug application, or BLA, biologics license application, or license application, BLA, which is between phase 3 and 4. Contents of NDA/BLA, clinical data, which is the data from phase 1 to 3 trials, manufacturing data, of course, detailed description of the IP's production process, labeling information, instructions for use, indications, and warnings. So there's the difference. So an IND, an investigational new drug application, IND, prior to phase 1, in order to get the approval to do phase 1 to 3, we need the manufacturing information, the preclinical data, and the study or the trial protocol. While here, we need the data for the BLA or NDA, which is between phase 3 and 4. We need the data from the trials phases 1 to 3, the phase 1, 2, and 3, the data from there. Of course, the manufacturing information still, and the instructions of using the drug, since it will be in the market, although it's phase 4, but in the market for the population, available for the population to be handled or after prescribed to the if it needs prescription to the population for the patients. Public patients or patients in public. So instructions, medications, instructions, or drug or labeling information. Example, after completing phase 3 trials for a cancer drug, the company submits an NDA, new drug application, to the FDA to request approval for marketing the drug. Now, phase 4 post-marketing surveillance. Once the IP is approved and marketed, it enters the post-marketing surveillance phase. Objective, monitor long-term safety and effectiveness in the general population. Activities here, adverse event reporting, monitoring and reporting side effects. Phase 4 trials, ongoing studies to gather additional data about the drugs used in diverse populations. Example, after approval, the company continues to collect data on the drug's long-term effects in patients and reports any serious adverse events, SAEs, to the regulatory body. Summary tables. Here we have a table on overview of IP development cycle, life cycle. Table on clinical trial phases and focus. Table on contents of NDA BLA/BLA submission, new drug application / BLA biologics license application submission. And of course, this applies into FDA. However, there are similar or parallel to these applications, similar same processing of these FDA applications worldwide, maybe with different titles or different names. for other regulatory bodies in other places in the world. For instance, worldwide. For instance, again, let's say MHRA, MHRA in United Kingdom or EMA in European Union, and so forth. Here's a table on post-marketing surveillance activities, table on key stages in the investigational product lifecycle. Next, we have the MCQs, multiple choice questions, in which we will revise what we have covered through this lecture. Question 1, what is the primary purpose of phase 1 clinical trials? Would be B, to determine dosage range and assess safety, as phase 1 trials focus on the safety, dosage, and side effects of the IP investigational product using a small group of healthy volunteers. Question 2, what is included in the contents of NDA submission? So NDA, new drug application, or BLA, biologics license application, in which is exactly in between phase 3 and 4. What we need, of course, the data from the trials or the phases of the trial 1 to 3, manufacturing information, and the labeling information. So would be B, clinical trial data from phase 1 to 3, manufacturing information and labeling information or info. As an NDA requires comprehensive information including trial data, production details, and labeling. And labeling information is exactly the medications or drugs instructions, how to use, or directions. Now, what if the question was, what is included in the contents of an IND, investigational new drug application, which is prior to phase 1? So we need the contents: preclinical data, this is studies that occurred or conducted on animals, preclinical data. Still, we need the manufacturing information, of course, and the study protocol. How the protocol for the study, how will be conducted on humans through phase 1 to phase 3. Question 3, in which phase are long-term safety and effectiveness monitored after market approval? Of course, phase 4 would be D. Phase 4 is the post-marketing phase, focusing on monitoring the IP's long-term safety and effectiveness. Question 4, which of the following best describes an investigational new drug application, IND? Would be B, a request to start clinical trials in humans starting for phase 1. So an IND is submitted to request permission to begin human trials after preclinical research. Question 5, what is the primary focus of phase 2 clinical trials? Will be efficacy in addition to further safety monitoring. So A, confirm efficacy and monitor side effects. So A would be the answer. Phase 2 trials aim to confirm the efficacy of the IP and assess its safety in patients with the targeted condition, not like phase 1, healthy volunteers with the targeted condition in phase 2. And here we come to end of this lecture. Thank you very much and goodbye.